All right, everyone, we'll go ahead and get started. Thank you so much for being here today. Today, we're going to tell you about something that we've been working on developing in-house that we're really excited about, which is the ability to use your photometry signal to control hardware using a Neurophotometrics FP3002 system. And before we get started, if you have any questions that come up at all during the talk, you can use the little Q&A button down at the bottom to submit questions, and then we will answer them at the end. So before we get into it, today we're going to be talking about how to do closed loop experiments with fiber photometry. And if you're not familiar with fiber photometry is an in vivo imaging technique that allows you to record neural activity or neurotransmitter release in real time with millisecond temporal resolution. And the type of activity that you're recording depends on the sensor that you use. And so the most common sensor that we use for fiber photometry is called GCamp, which is a calcium indicator. And this is useful because when a cell fires, calcium rushes into the cell, and then we can look at those calcium transients as a proxy for neuronal activity. And so there's lots of different types of variants of sensors, but there's one common theme amongst all the sensors that are used for photometry, and that is that they have some resting basal fluorescence, and then upon binding to their molecule of interest, their capacity to fluoresce increases by orders of magnitude. And so what we could do is then inject these sensors into the brain, into our region of interest. You can also isolate specific cell types by using transgenic animals. And with this approach, we can then measure those changes in fluorescence as a proxy for neuronal activity or neurotransmitter release. And so what you get is kind of an average of the activity of all of the cells that are underneath your fiber. And it looks like something like this, where you have these peaks in your signal that correspond with neural activity. And so there's lots of different ways that you can set up your fiber photometry experiments depending on how you express your sensors. So the most common is to record from cell bodies in some region of interest. But you can also pick up signal from terminals if you're interested in looking at projections to certain regions or isolating that signal in various projection targets. Using some of the novel biosensors that bind to specific neurotransmitters, you can also measure extracellular neurotransmitter release with this technique. So it's a highly flexible technique that can be applied to a wide range of experiments. And so what we're going to be talking about today is how to incorporate closed loop experiments in with your fiber photometry experiments. So before we kind of get into the nitty gritty, I just want to stop and talk about what a closed loop experiment is and what this means. So closed loop systems, by definition, are automated control systems in which an operation or process is regulated by feedback without the need for user input. And so kind of the most simple example of a closed loop system is a thermostat. And so if we think about a thermostat, you have a thermometer that's sampling the temperature of the air, and then you set some threshold of temperature for which if the temperature exceeds this set point, the thermostat will turn on the AC. So this information gets fed to the thermostat controller, which then decides whether or not to turn on the air conditioning unit, which then changes the air temperature and goes back and alters the temperature read by the thermometer. And so this is kind of the easiest example of how a closed loop system would work. And so in order to adapt this for neuroscience, we can kind of rename these terms with more general words. So in neuroscience, you have some kind of data stream, some kind of measure that you're monitoring in real time, and then some kind of threshold or criterion that that data must meet in order to execute an action. And then you have a controller that is tracking all this information and deciding when to actually trigger hardware output or not. And then you have an actuator, which is your hardware that's actually acting on the system. And so what does this look like in the context of a neuroscience experiment? So I'm going to explain this kind of with an example from the literature because I think that's the easiest way to make sense of it. So this is an example of activity recorded from a discrete population of granule cells in the dentate gyrus of the hippocampus during seizure. And so in this paper, what they showed was after kindling of a seizure, there's a delay in the signal response, and then they get this really nice upswing followed by this increase in activity. And so say that you're in this lab and you're wanting to see, okay, if I optogenetically inhibit these cells, can I interrupt this process and stop the seizure? So what type of experiment would you normally do next? So one really common thing that people do is optogenetic stimulation that's time locked to some event, normally, for example, the kindling of the seizure. And so if you were to set your experiment up this way, what you can see is that you're going to be delivering inhibition during all these different phases of the signal, right? There's lots of changes that are happening as we're delivering our inhibition. But what if it turns out that the part of the signal that's responsible for driving seizure is just the second peak? How can you then make your experiment more precise to just target the part of the signal that you think is responsible for driving your behavior of interest? One way to do this is using closed loop experiments. So say, for example, instead of just inhibiting for some window of time across all of your animals, you can identify certain signatures in the signal and use that signature to then control your laser to deliver really precise time-locked optogenetic inhibition. And so there's a couple key advantages of this approach. First, that you get much better temporal precision. And this is something that comes up quite a lot in fiber photometry because every signal is going to look different and every mouse is going to look different. And so if you're doing things based on the signal within that exact mouse in that exact moment, you get a lot more precision in your manipulation. And as a result of this, you can reduce the off-target effects of your manipulation. Another key benefit of this is that you can adjust your intervention in real time based on the signal. So say, for example, you start to deliver your inhibition, your signal goes flat, but if you had like a fixed window of inhibition, you're going to keep delivering laser inhibition, which might then give you some kind of off-target effect. However, if you're tethering that inhibition to the signal itself, that gives you the advantage of being able to adjust in real time and turn that laser off. And so this is a really nice example of how closed-loop experiments can provide a very elegant and time-locked way to probe the relationship between brain activity and behavior. Because what we know from many, many neuroscience studies is that this relationship is very closely time-locked. And if you're off by a matter of seconds or milliseconds, you might not be doing what you think you're doing. So this temporal precision is really key. So in neuroscience, there's a lot of different data streams that you could use to drive in a closed loop system. So one of the most common is animal tracking. I think this is something that many labs do already. But with our system specifically, you can look at different features of the animal tracking to control your closed loop system. So you could use the location or the velocity, and the criteria that you would set for these parameters are perhaps that the mouse is in a certain zone or their velocity is over certain thresholds. You can include that they're like darting or freezing or anything like that. What we're going to be talking about in more detail today is how to actually use your photometry signal to then control your hardware. So with this, there's multiple measures that you can look at, one of which is just the average signal, or if you have a signal that's quite peaky, we can do real-time peak finding and use those peaks then to control hardware. But this is really flexible, and the benefit of our acquisition software, which is Bonsai, is that you can interface lots of different hardware with Bonsai. And so with just depending on the way that you configure your workflow, you could use almost anything as your data stream. So if you connected some kind of lever or something with when you're measuring like beam breaks for tracking or a nose poke port, you can then use the events from this external hardware to then trigger, for example, the laser within the system. So it's quite flexible in terms of how you can set your experiments up. In terms of actuators, there's lots of options as well. So the most common when you're using the photometry system is either that you're driving the lasers for optogenetic stimulation or inhibition, or you can control data acquisition. So say, for example, you're doing really long recordings and you only want to record data when the mouse is like awake and in some, portion of the environment. You can then use the tracking to only trigger recording when the mouse is in your zone of interest, and it would turn off when they exit. And in terms of hardware, you can also essentially use anything that can connect either directly to Bonsai via an Arduino, or anything that can be triggered via a TTL pulse using the two digital outputs on the system. So with this kind of setup, you could perhaps use brain signal to control the delivery of a reward or the presentation of some kind of cue. You could do electrical stimulation, you could use an infusion pump to infuse some kind of drug, or some kind of behavior camera. So it's quite flexible in terms of the types of experiments that you can do. And just to mention, because this is something that I kind of skipped over, in this case, with our photometry system, the controller is always going to be Bonsai, which is our acquisition software. So just to kind of give you a sense of what these types of experiments look like, this is an example where we're using tracking to turn on optogenetic stimulation. So in this example, we're doing some real-time animal tracking, and then we've defined this zone here around this food hopper up in the top left corner. And what Bonsai is doing is identifying when the mouse is in that chamber or not, and spitting out some kind of Boolean value, either true or false. True for mouse is in zone, false for mouse is not in zone. And then what you can do is take this information and use it to trigger your actuator of interest. So what we're most excited about today is kind of explaining how you can use your photometry signal to trigger output, because this is something that's really quite new in the field, and something that we've spent a lot of time putting effort into developing. And so let's talk a little bit about what this looks like and how it works. So essentially, when you use your photometry data as your data stream, you can use brain activity to control hardware. And so when we think about photometry signals, every single one is different, depending on the cells that you're recording from, part of the brain you're recording from, what sensor you're using. The signal is going to look different based on all of those parameters. And this is just an example of of the variety of responses you could get. So all of these recordings were done in the exact same population of neurons in the same mice, with all the same parameters. And what you can see is that the shape and the kinetics of the response are very dependent on the type of behavior that the mice are performing. And so this really nicely demonstrates how there's kind of a variety of ways in which you might want to look at your signal. And it's going to just depend a lot on your exact setup. And so an issue that we kind of came up against in development of these workflows is that there's really not a one-size-fits-all solution when it comes to doing some kind of real-time analysis with your signal. And so what we've done is develop two different approaches and two different methods for analyzing your signal. So the first is peak detection. And so with this workflow, essentially what you can do is recognize peaks in your signal in real time. And so this is good for experiments where you have a really spiky population, or maybe you're using a fast variant of GCamp where you're getting individual spikes. that are pretty well resolved. What you can do is detect these spikes in real time and use those spikes to control hardware. And so the output from the controller to the hardware in this case would be a Boolean value, either true or false, peak is occurring or peak is not occurring. If you have a population where you don't really have spikes, kind of akin to these second two traces up top, or you have a response that's less of a peak and more of kind of a slow shift in baseline. And this is something that we see often if you're recording from terminals or if you're using some of the novel neurotransmitter sensors that aren't necessarily peaky. What you can look at instead is the average signal. And so with this workflow, essentially what we do is we calculate the average signal over some user-specified moving window, and then the output that's getting sent to hardware can either be continuous or Boolean. So if you want like an all or nothing event, like for example, when the brain activity reaches a certain threshold, you want to deliver a reward, that's possible. But with this workflow, it's also possible to use a continuous measure. So say, for example, you're controlling like the volume of a white noise generator, and you want the volume of that sound to match the brain activity. In that case, you could feed some kind of continuous value to your hardware. So we have lots of options to kind of capture the different features that you might see in your photometry signal. And so let's pivot and kind of talk about how this actually works. So when we're using brain signal to control hardware, it requires us to do some real-time analysis. And so one thing that's really common in photometry experiments is that you get kind of a slow decay shape in your signal from photo bleaching. And if we were to take those numbers and use them to control hardware directly, what you would get is a slow decrease where, nothing ever happens. And so what we need to do is first correct out that shape. And the way that we can do that is using a built-in control. So our system has a handy built-in control that takes advantage of an optical property of GCamp that allows us to identify the shape of that curve from photo bleaching and then use that to correct our signal. So like I mentioned before, GCamp has some resting fluorescence, and when it binds to calcium, that fluorescence increases by orders of magnitude. And so if we look at the excitation curves for these two states of GCamp, what we can see is for unbound GCamp, the relative fluorescence emission is pretty low, but when it's bound to calcium, it increases greatly. And the important thing to note is that around 420 or 425 nanometers, these two curves cross. And so what that means is that if you stimulate at that wavelength, any fluctuation that you get in your signal, you could attribute to something other than an actual event. And so what we can do is record data at both 470 nanometers, which is typically what we use for most green shifted sensors, as well as 415 nanometers, which gives us a motion control. And so the way that we think of this is that if you have motion artifacts in your signal, or you have a decay shape from bleaching of autofluorescence in the tissue or the patch cord, or you have some kind of artifact from the patch cord bending, that's going to show up in both the 470 and 415 signals. However, or really theoretically, most of your neural activity is going to be captured exclusively with your 470 channel. And so what we can do is then remove the 415 contribution to the signal. And what that allows us to do putatively is isolate our calcium dependent signal. And so this is what these traces typically look like. And so what you can see is that we have this kind of slow bleaching shape in both of our signals. And then in our 470 channel, we have all kinds of dynamics on top of that slow decay that we can attribute to our actual neural activity. And so if you record with these two wavelengths, what you can do is then use your isospastic signal. And it's called the isospastic because this point where the two curves cross is called the isospastic point. We can use these data to then flatten out and isolate our actual neural activity. And so let me show you what this looks like in real time. So just for reference, this is kind of what our full traces look like. So typically, when you're doing some kind of signal analysis, you're doing it post hoc. But because we want to use brain activity in real time to control hardware, we have to do some real-time analysis, which looks a little bit different. So if you were doing this type of analysis post hoc, typically what you would do is fit a curve to your 415 data, scale that fit to your 470, and then subtract out that fit to flatten your data. However, when we're doing this in real time over a moving window, we don't fully get a bi-exponential decay like we would on a full trace. And so what we found is that if you just take a linear fit over these smaller windows, it works just the same. So the processing steps are as follows. We take a linear fit of our 415 data. We scale that to our 470 data because we assume that the rate of photo bleaching is the same between the two channels. We can then subtract that scaled fit from our raw 470 signal, which will then give us a flattened signal. And then once we do that, we normalize our data within that window. So if you were doing an experiment where you were looking at your average signal, all you have to do is then average over your moving window, and that's the value that you feed into your hardware. But the example that we're going to talk about today goes one step further, and that's via peak finding. So this was something that we spent a lot of time developing, and the results were quite exciting. So when you're doing peak finding post hoc, the steps are a little bit different because you know both when the peak goes up and when it comes down. However, if you're doing this in real time, you don't have time to wait for that peak to go down because it's going to introduce a significant delay between neural activity and hardware execution. And so we had to get a little bit creative to figure out a way to identify those peaks as soon as that peak starts to rise. And so what we figured out is that if you separate out your signal from noise, and there's always going to be noise in your photometry signal, you can remove this noise or really estimate your signal with no noise by passing your raw signal through a smoothing filter. And so what this is going to do is give you an estimation of your noiseless signal. And so if we kind of zoom in a little bit closer, what you can see is that our smooth signal follows kind of the slower fluctuations in the signal. And what we've done is remove the noise on top of that. And so if we then subtract our smoothed fit from our raw data, we can extract just the noise. And so what we noticed was that during the rising edge of a peak and really the falling edge as well, you get this really big change in the noise where it goes from being quite large to a lot smaller. And so what we figured out is that you can actually identify when peaks are occurring by just looking at the mean absolute deviation of the noise. And so what we do is we take the MAD of the noise over the same rolling window that you're using for all of your other analysis, and we generate what we call an indicator value. And so what this indicator is just the instantaneous MAD of the noise, and you can set a threshold for what you want to consider a peak. And so what we saw is that this value drops during the rising edge of a peak. And so a lower indicator indicates a higher likelihood of a peak occurring. And so what you can do is then set your threshold for what you're counting as a peak. And so this is what this looks like in real time. It's quite fast. And it's essentially near instantaneous. So the really important thing about this is that it's not introducing a delay into your signal, which is so important in neuroscience because neurons communicate on the scale of milliseconds. And if you're detecting your peak 500 milliseconds after it's occurring, it's too late. And so this is something that we're quite proud of. And there was a lot of engineering that went into the system in order to make this possible. And the first is that we spent a lot of time figuring out how to make exceptionally stable light sources, because if you have any fluctuation in your excitation light, you're going to have fluctuation in your signal as well. And so we spent a lot of time to try to figure out how to make this as stable as possible so that you are confident that any fluctuation in your signal is due to actual biological activity. Another thing that we did was integrate photodiodes into the excitation light path. So if there were to be any shift at all in the brightness, there's kind of integrated feedback within the system that would then adjust your LED power so that the light that's actually getting delivered to the brain is very stable. We also have our built-in 415 control channel that allows us to separate out those artifacts from our actual signal and accurately detrend our data. And we also spent a long time optimizing the settings of a CMOS camera sensor, which is what's actually detecting the light coming back from the brain. And this is really important because it allows us to pick up those tiny changes in the noise that are indicative of whether or not a peak is occurring. If we couldn't see the noise at all, or if we couldn't distinguish the size of the noise, none of this would be possible. We also have quite flexible acquisition software that allows you to add your own Python code to your workflow to do whatever type of analysis you want, and also makes it possible to interface with all types of different hardware. And then important for this example today is that we also have integrated lasers. So you can take this brain signal, send that signal to the laser, and you can stimulate that exact same region of the brain that you're recording from to modulate activity in real time. So next, what I want to do is highlight what this looks like in real time. So I'm going to show you a case study example where we're using peak finding to control optogenetic stimulation. So for this workflow, the data stream is the FB signal, and in this case, we're looking at peaks. Our threshold is a cutoff that we've determined for our indicator that I showed you earlier. This information is getting fed into our Bonsai workflow, which is then determining when to trigger a laser for optogenetic stimulation. And so I am going to show you what this looks like in real time.
Hello. Today I will showcase a closed-loop fiber photometry workflow that uses peaks in the activity data to trigger optostimulation. This workflow does a real-time analysis of the activity data using a peak finding algorithm. Then, when a peak is found, the workflow triggers a laser pulse train. Currently, I'm using this workflow to detect peaks in a calcium-dependent signal. I opened the visualizers of some key nodes to show how this workflow is operating. First, we have the photometry data visualizer, which shows that we're recording from a green channel region of interest using a 415 nanometer and the 470 nanometer LEDs. This gives us two signals, the isospastic and the calcium-dependent signals. During the analysis of the isospastic and calcium-dependent signals, we generate an indicator that can be used to determine whether a peak is occurring or not. That's what's shown in this visualizer. This indicator is inversely related to the prominence of the peak. So higher indicator values imply that there's less likely to be a peak, while lower indicator values imply that there's more likely to be a peak. We then use an indicator cutoff value to determine if a peak is present, and that is shown in here. So right now there are no peaks present, but soon this will turn high, which means it detected a peak, which is shown here. Once we have a true or false value determining whether a peak is present, we add some additional logic for determining whether optostimulation should be started. In this example, when a peak is detected, the logic starts a 1 second duration optostimulation pulse train. This can be seen in the generate optostate visualizer. We conclude this workflow by passing the optostate signal back to the beginning of the workflow, to generate start and stop stimulation commands to be sent to the FP3002 node. You can see the laser was successfully triggered by noting that this digital I/O state alternates from true and false, while this generate opto state is 1. Hello.
Okay, so like I mentioned before, there is no one-size-fits-all analysis method that works for every single photometry experiment. And so what we've done with this workflow is build in a couple of user-defined parameters that allow you to adjust how the peak finding is working and tailor it to your signal. And so in this workflow, we've built in two different parameters, which are buffer duration and indicator cutoff. So the buffer duration is the duration of the moving window over which you're doing your analysis. And then the indicator cutoff is where you set the threshold for what gets counted as a peak and what doesn't count as a peak. And so it's quite complicated to try to figure out exactly what the best parameters are for your signal. And so what we've done to make this easier is build a tool to help you figure out those parameters. And so what we recommend doing first, if you're interested in implementing this in your experiments, is first record a baseline signal. If you can put the mouse in the environment in which you're testing, that's ideal. If you're going to test in a novel environment and you need to get the parameters dialed in a different environment, that's fine too. But we recommend recording, maybe 10 or 15 minutes of just activity so that you can characterize the signal. And so what we've done is build a Jupyter notebook with essentially all of the same code that's in the workflow so that you can test post hoc and figure out your parameters. And so these steps are exactly what I showed you before, except instead of doing this on a rolling window in this analysis, you're kind of doing it on the entire signal. And so I won't go too much into this today, but essentially how it works is you put in your signal and you detect peaks using kind of traditional post hoc peak detection methods. And then you follow the same analysis steps as you would in real time in the workflow, and you can kind of change the parameters for your window size and your indicator to see how well your real-time detected peaks line up with your post hoc detected peaks. And that allows you to then dial in your parameters so that you're detecting peaks that you would consider peaks. You're not getting extraneous noise that's getting counted. And you're not missing peaks either. And so after you kind of go through this tool and figure out what those parameters are, you can then apply them back to your workflow. And so I have an example of what this looks like live.
This workflow requires some calibration to be successful. In particular, the buffer duration property and the indicator cutoff property need to match the type of signal that you're working with. For example, if you're expecting short duration peaks, you might not want a large window size that can contain many peaks at a time. Or if you're expecting sharper, more prominent peaks, you might consider having a lower, more restrictive indicator cutoff value. If you do not know what to expect, it's best to characterize your signal a bit before running this workflow. To do this, run a control trial with the standard photometry workflow and the FP3002 configured in the same way that it will be for your experiment. We can easily do that with this current workflow by selecting all the nodes other than the standard photometry nodes and clicking Ctrl D to disable them all. Now we're left with just the FP3002, the photometry data, and the photometry writer node, which will give us our control data. Then you can go ahead and run the workflow for around 10 to 15 minutes to get some good control data. Once you acquire that data, you can go ahead and stop the workflow, and you can go over to your post hoc analysis. After conducting a post hoc analysis, we find the normalized detrended signal here in the bottom figure. And then we did some post hoc peak finding as well to find the relevant peaks in our data. And we want to see how our real-time analysis matches up with this. So in the top figure, we plot the indicator value that would have been calculated from each photometry data frame over a certain window size and we want to see if our indicator would have detected a peak or not. So this particular plot has a window size of 3 seconds, and you can see it's fairly noisy, where some of the more prominent peaks, here, here, and here, are successfully detected if we have a low enough indicator cutoff value. But it would not successfully detect a lot of the smaller peaks, because if you increase the indicator cutoff value enough, you're going to get some false peaks, such as right here, where you have a local minima in your indicator, which would indicate a peak, but there's no actual peak detected in our post hoc analysis. So what we want to do here is we want to scan our window size to find the best window size where we can have the most peaks aligned with local minima in the indicator without having extraneous peaks detected that weren't actually there. So we did this for window sizes from 3 to 25 seconds. And after looking through these plots, we determined that 9 seconds was most likely the best case for this signal. At 9 seconds, we found that the 3 main peaks were easily detected with fairly low cutoff values. So the next step is to find the best indicator cutoff value. Here we scan from 0.001 all the way up to 0.035 to find the best cutoff value that's going to detect the majority of peaks without introducing a bunch of false positives. For example, if you select like 0.03, then most of the time it's going to be detecting a peak, but if you select 0.005, you're only going to be detecting these largest peaks. We want somewhere in the middle where we can detect some of the smaller peaks without introducing a bunch of false positives. Ultimately, we decided a window size of 9 with an indicator cutoff of 0.01 was best for this signal. Here we detect the 3 largest peaks as well as some of the medium-sized ones. An example of a false positive is right here. So in our normalized detrended signal, there's no peak here. But in our indicator, we see that there's a large drop here. And so if we increase our indicator cutoff to include this drop right here, then we're going to introduce a false positive where there's no actual peak. So once you determine the best window size and indicator cutoff value, you can go ahead and come back to your workflow and re-enable everything. I'll also reopen the visualizers I had before. So we had the indicator, or the get indicator one showing its plot. We had the is peak present plot as well. And we had this generate optostate plot. Since we found the ideal window size and indicator cutoff value for this type of signal, we can now update the real-time analysis node with those values. First is our buffer duration, which is going to equal our window size. And we calculated the best window size to be 9 seconds. Next is our indicator cutoff value. And we found that 0.01 was sufficient to detect the majority of the peaks without introducing any false positives. With our real-time analysis configured, we can now configure our FP3002 system. We can do this by double-clicking the FP3002 node to open its editor. Our first tab is our mission alignment tab, where we can draw our region of interest. Here I will draw a green channel region of interest. Next is our excitation sequence, and we configured this workflow to work with calcium-dependent signal and an isospecistic signal. So we do not need this 560 nanometer LED. Instead, we're just going to alternate between a 470 and a 415 nanometer. That should give us 2 signals. Our 415 nanometer will give us our isospecistic, while our 470 nanometer will give us our calcium-dependent. Next, during our control, we recorded at 40 Hertz, so let's make sure we match that so our real-time analysis configuration is still sufficient. So I'll enter 40 Hertz into our frame rate property. Next is our excitation power, and we had this at 5% for both LEDs. And finally, let's configure our opto-stimulation tab. First is our wavelength. We're going to use a 635 nanometer laser. Then our amplitude, we'll go ahead and do 10%. And our frequency will be 100 Hertz. And we'll go ahead and do a 50% duty cycle So we'll have a pulse width of 5 milliseconds. Now this pulse train type has to be a continuous pulse train type because we're controlling the pulse train duration using the workflow. So normally, if this is set to finite, you're controlling your pulse train duration by this pulse count property. So if I do 15, it's a longer pulse train duration. However, we've allowed the workflow to take over this logic for us. So we just set this to continuous here, and then in the workflow, we'll set our pulse train duration. So with the laser configured, we can go ahead and go to the opto or digital IOs tab. And the only change we need to do here is we need to make sure that this digital output 1 is set to both. This will allow us to control the internal laser while also using the BNC port to timestamp when the laser turns on and off. With that, our FP3002 system is configured, so we can go ahead and close this, and we can go to our opto stimulation logic and set our pulse train duration. Last time we had it at 1 second. Let's go ahead and change this to 3 seconds so that we have a 3-second pulse train. Now that we've configured our workflow, we can go ahead and run it. Now these 3 nodes are going to take a little bit longer to populate because we have a longer buffer duration. Our buffer needs to fully populate before we run our real-time analysis, and so it's going to take a couple frames to start our real-time analysis. An important note is that although we have a longer buffer duration than before, we have not induced any more latency to the detection of peaks. When a peak occurs, we will still nearly instantaneously detect it and then trigger opto-stimulation accordingly. It appears that we've detected a couple peaks and subsequently triggered opto-stimulation. So that concludes the calibration portion of this workflow.
Great. So I know everybody is sitting here looking at that workflow, trying to figure out how you're going to build it. You don't have to. If you're interested in trying this out in your lab and you have an FP3002 system, let us know. Send us an email at support@neurophotometrics.com. Let us know that you're interested in trying this, and we can send you the workflow, all of the documentation and materials, as well as that Jupyter notebook where you can do your post hoc analysis to try to configure your parameters. So if you're interested, please send an email to us here and we'll get those materials to you. Thank you all for being here today. If you have any questions that come up after the webinar, feel free to send us an email. You can also check out our forums on our website. And if you have questions now, you can use the Q&A button. And then after this webinar, you'll receive a follow-up survey. If you can fill that out, that would be super helpful to us. And if you're interested in learning about other MBF products, we have all kinds of upcoming webinars that we will send out emails for, and they'll be on our website as well. So keep an eye out for those, and we'll take any questions.
Olivia, I've been answering some of the questions in the chat, but one question that was posed was, does the buffer size or the window size you use affect the feedback delay? And if not, why not?
Chris, you want to take that one?
Yeah, I can take that one. So the answer is no, it doesn't affect the latency of detecting a peak, and that's because of the type of buffer that we're doing. So we're doing like a running buffer, so like as one photometry data frame comes in, one leads. So if your next photometry data frame is part of that peak, you're going to see a massive spike. And so that's going to be found using the indicator, and it's going to be a massive drop. So in some of the plots we showed, we saw like as soon as the peak occurred, it just drops super low and it stays low. So when you're increasing your buffer duration, you're more increasing that tail length, or like how long is the peak detected for, because the peak's going to be in that buffer for like 9 seconds or 30 seconds. Does that answer the question?
Yeah, that's fantastic. One question we got was, does this work with the first generation system or the B1 system?
Yeah, I can take that one too. So the first time we did this was actually on a B1. So that was where we developed the algorithm first. And then we improved it and made it available for V2 as well. So you just make a couple tiny tweaks in the code and it'll work for both systems.
And I'll also do a shameless plug that we do have a really economical upgrade promotion going on. This type of real-time processing does require an incredibly stable signal, or it's very much facilitated by a really stable high SNR recording. And the V2 compared to the V1 has, at least a 10x increase in SNR. Couple questions. So for signals that peak but have a slow, I guess with Gcamp, the kinetics are asymmetric typically, like a faster rise and a slower decay. Is there a mechanism you guys could build to detect the end of a peak instead of the beginning of a peak?
That's a good question. What we've seen is that the noise gets much smaller on both like the rising and the falling edge. So there's probably a way to tweak the workflow where it's like looking for the transition from like low noise to high. We haven't tested it yet, but I think it's possible.
So, and also for smaller, we found that with smaller window sizes, it can pick up the beginning and the tail, and not like the very middle of the peak if it's like smaller than your peak duration. And so if you add a little bit more logic there, you can say, all right, make sure like the slope is negative, so it's decreasing, not increasing. So you can isolate those out.
Yeah, another option would be adding a bit of logic after this, because we do see when we detect the peak, and then when we stop detecting the peak is very close to the end of the peak. So the nice thing about these types of real-time processing algorithms is that they're easily stackable if you have to have other constraints. One question was, how does this work with indicators that aren't Gcamp? So like grab DA or grab norepinephrine. Are there things that you'd have to tweak? I know the isospecific signals can be a little different for those.
Yeah, so it would just depend on what your signal looks like. Some indicators tend to be quite peaky, like blue sensors, dopamine can be quite peaky, and some of them are really slow where you get more of that baseline shift. And so I think between the peak finding and the average signal, one of them should be able to work for your experiments. With the difference in isospecific point, that's a little bit more tricky. Because often the isospecific point for most sensors isn't published, and so we don't have a lot of great information. But there are other ways that you can do controls to correct for photo bleaching. And so if you're doing an experiment where maybe you're like co-injecting a TD tomato or an M cherry just as a static indicator, you can use that channel as your like baseline signal. You don't necessarily have to use the 415. And so that would be one way to get around the isospecific if you have a sensor where there's a lot of overlap in the 415 and the 470. Another option is to actually like fit the 470 signal to itself and like flatten out that way. It's a little trickier because if you have a really big event that occurs within that window, then you can skew your signal a little bit. But if you make your buffer size long enough, you should still be able to do it that way. So I think whatever controls you use, it's possible to modify the workflow to then use that to flatten out your signal.
So another question, is it possible to detect sensor activity in one region and opto-stimulate in another region if you're doing multi-site recording? Are you confined to only stimulating the region you're recording from?
Yeah, that's a great question. You can stimulate in a different region. It just depends on where you focus your laser. So if you're recording from one ROI and then you focus your laser down a different patchboard branch, then you could do stimulation in another region that way.
Awesome. Yeah, the one thing that you do want to be a little careful of with these types of experiments is if you're doing like, you know, non-specific kind of cation channel, like channel rhodopsin or crimson, the stimulation could induce a peak and you could accidentally create a positive feedback loop between your setup. So it is important to do some titration experiments to see the effect of, like, what effect does stimulation, say at 1 millisecond at, you know, X laser power have on your signal. And you might want to blank out some of those areas from your closed loop setup. I guess this is kind of a similar question, but if you have multiple regions of interest, could you have a setup where you only stimulate if you see a peak in both regions?
Yeah, you can do the same real-time analysis on n different regions, and after that, you can use whatever logic you want. So you can say like an OR gate, where like if one of them is indicating there's a peak, then stimulate, or you can have like an AND gate where like both have to be indicating a peak is present, and then you do your optostim. And we've done also similar things with the like the average signal instead of the peak finding, where we averaged across the population and use that to control hardware instead of just a single region and a single animal.
Cool. So the peak detection is kind of digitizing the signal because it's an all or none. Have you guys had experience working on things that are more scalar? Like if you are looking at the baseline, and can you have that affect some sort of actuator or output that is not binary?
Yeah, we've done this for labs before. So if it depends on the hardware that you're using, and if you have some kind of hardware that can accept a continuous value, then you can feed it that value. And you actually can do this with the peak finding workflow as well. You don't have to just do like all or nothing for peaks, but you can, same thing, just take like, it's a average over the moving window. But yeah, you can essentially like, you know, control the pitch on a tone generator and have it go up and down with brain activity or something like that.
Very cool. Any more questions from the audience?
I did see one about the recording. So this recording should go up on the MBF website. It may take us a couple days, but you should be able to access it on there. Actually, I will go back to the slide with the emails. Yeah, if you have any other questions or if you would like this workflow, please send us an email, or you can visit the forums on our MBF site.
Is where could I see this photometry system in action, Olivia?
Oh, so we will be at Monitoring Molecules May 13th through 16th in Chapel Hill. So if you're going to be at that conference, we'll be there. We will also be at GRC Optogenetics this summer in Italy. We probably won't have a system with us, but we'll have some data on a poster, and we're also happy to chat. And then we'll be at SFN as well in October in Chicago.
So we do have one other technical question. Is it possible that the linear fit that you guys are kind of estimating from the isospecific signal may affect normalization of the peak? Sorry, I might have gotten that question a little wrong. If Syed, if you could maybe clarify a little bit more. But are there any issues, I guess, that you guys could see with this linear fit? Like say there was a big motion artifact there or something.
Yeah, this is something that's really important to consider. It's a really good question. So if you have a really wide peak and your window is smaller than the width of your peak, then you can get some really funky signals. So that's where the calibration is really important, to make sure that you're setting your window size appropriately so that you're not like cutting your peaks in half because your window is too short.
Very cool. Any other questions? We got a couple minutes. I guess on the optostim, how flexible is your ability to control the various parameters of optostim? Like, can you kind of dynamically control power and frequency, pulse width?
Yeah, you can control the amplitude, the frequency, and the pulse width all dynamically within the workflow. So if you have, instead of doing like a binary true or false, whether a peak is occurring, if you instead have an indicator that's analog, then you can pass this through some Python logic to generate your target frequency or pulse width or amplitude, and then we can send that command to the opto stimulation instead of like start opto stimulation. So yes, you can dynamically change your pulse train while it's running based on your indicator.
Kind of in line with that, I know personally, and then a lot of folks in the audience who have done optostim, especially in the earlier days, the typical way that we decided on stimulation parameters was educated guessing. Typically, it was 10-ish hertz, 1 to 5 millisecond pulse width at like 10-ish milliwatts. Is there a workflow or a simple way to kind of scan that parameter space to make sure that the way that we're stimulating the animal is affecting activity kind of in the biological range that we're seeing, and we're not driving it out of the biological range?
Yeah, that's a really good question, and something that I think is kind of ignored a little bit in the opto field. We don't have something built currently, but there is, we actually that's not true. We have a workflow where you can like dynamically modulate the laser parameters. So what you could do is like do a test experiment where you stimulate with like XYZ parameters, and then you wait a minute, and then you stimulate with your other parameters and wait a minute. And then what you could do post hoc is see like what stimulation parameters give you the desired result that you want. I think it would be something that's difficult to do in real time. But certainly like you could just kind of scan through parameters and see which ones are giving you your desired effect.
Awesome. Well, I think you guys have answered all the questions, and yeah, I want to thank you guys. This is really fantastic. I know a ton of work went into both making the webinar as well as designing the algorithms underlying this process, and I know we're very excited to see people start to do these experiments. So anyone in the audience, feel free to reach out. We're happy to go over things in more detail offline. With that, have a good rest of the day.
Thanks, everyone.
Thank you.